Yes, Monica.
Good morning, everyone. I am Monica Kaushik, and I'm one of the members of the organizing committee for the Bird Monitoring Symposium. I'm a faculty of biology at the Azim Premji University, Bangalore, where I find ways to slip bird in every conversation and examples in my classes. So you can imagine the agony of those students who are interested in other texts. But that's my secret way of creating this army of bird enthusiasts and researchers who will go in the world and watch bird and probably do some research on birds. I welcome you all to the third symposium on bird monitoring in India. We are officially 3-year-old now and still learning to fly and developing our wings. We have received tremendous support from the bird watcher and researcher community over the past 2 years in form of presenting their work, giving feedback, and volunteering for organizing the symposium. It's the ever-increasing size of our community that is keeping us motivated and excited to come up with interesting topic every year. Your presence here today is a testament that the birding community is devoted to keep an eye on our winged sentinel. However, the idea of this symposium is to reach out to every possible stakeholder to underscore the value of long-term monitoring of birds across different habitats in India. And working together, we can make a stronger case for monitoring and evidence-based conservation of birds in India. Over the past 2 symposiums, we have learned a lot about monitoring effort of varied sizes and got introduced to relatively shy and elusive individuals and organizations who are involved in monitoring. Bird Monitoring India teams hope that this platform allows researchers, bird watchers, citizen scientists to know each other, support each other's work, and hopefully work together. Through this symposium, we expect that the collaborations will develop organically. All these stakeholders that I've just mentioned would come together and work toward this larger goal of building a long-term monitoring network for birds in India. And we can play our respective roles for the larger goal. Researcher involved in monitoring bird can help in identifying the species-specific and location-specific threats to the bird, highlight the challenges and limitations that they are facing at different stages of monitoring, and suggest material for support that they require for improving their bird monitoring work. On the other hand, we seek managers to actually tell us what exactly on ground are those questions that they want answers on, what are those ecological phenomena that they need researchers to come up with monitoring tools or a monitoring protocol. And most importantly, I think the bird watchers have a major role to play for monitoring. They are the ones who are actually going out and watching birds from various places, from their backyard and balconies, and feeding this information into the platforms such as eBird and eBird India. In the past 2 symposiums, we have had a whooping 1,742 people participating over different platforms. So our presentations and talks are streamed on YouTube, whereas discussions among fellow participants, speakers, all happens on Discord. And these people have been, the participants have been using these platforms for discussions. Over the past 2 symposiums, we also had 36 invited talks. We are particularly excited about increasing number of poster presentations that rose from humbling 16 in the first year to 26 in the year 2022. And this year we have 25 poster presentations. Concluding day of the first symposium included a discussion on future of bird monitoring studies in India. That also sowed the seed of for our organizing team to curate a list of tools, methods, questions that our monitoring community should ponder upon. That helped us to think of a structure of last and this year's symposium. In year 2022, there were 2 mini symposia, one on the use of radio telemetry for bird monitoring and second on bird atlases. Both these mini symposias were followed by moderated group discussion where participants as well as speakers from these mini symposias participated and discussed the challenges of these tools that they have been using for monitoring bird. Both these mini symposia included talks from researchers sharing their work, covering a wide variety of habitats, from trans-Himalayan landscape to the deserts of Kutch, and equally varied bird species such as great Indian bustard, white-rumped vultures, pied cuckoos, water birds, and hornbills, black kites, to name a few. If you're feeling, if you have this FOMO, which I generally have, if I miss an interesting symposia or a conference, so I would like to inform you that you shouldn't feel that way. All these talks are curated and are available on the birdmonitoring.in link. So you can go to the past event and look through all these talks. These mini symposias, as I was telling you earlier, were followed by focus group discussion involving researcher and participant thinking more critically about the important questions pertaining to the field of telemetry and bird analysis. Last year, we had two keynote speakers on each day, one on each day, Ms. Neha Sinha and Dr. Ghazala Shahabuddin, although they come from different professions, but both advocated for bird monitoring and its value for policy making. And coming to this year we have an exciting set of events lined up for 2 days. We have a keynote speaker on each day, but unlike last year, the keynote talks would be in the evening, as our speakers are from outside India. The symposium on the first day will shed light on the role of passive acoustic monitoring passive acoustic monitoring for bird. This is one tool that is increasingly becoming popular due to its USP of being less intrusive. However, unique tools come with unique challenges. So speaker of the symposium will share their work, followed by a moderated group discussion on the opportunities and challenges of this method. The second mini symposium explores the value of bird resurvey, which is more like the saying of standing on the shoulders of giant. So here, researchers are actually tracing the tracks that were taken by bird researchers, natural historians, in some cases centuries ago, to see what are the similarities and dissimilarities in the bird community composition, distribution, numbers, if any, in the recent time. And cherries on the cake are the poster session on both days by our participant, showcasing bird research and monitoring effort. And with this, I conclude and welcome you again to the third Bird Monitoring Symposium. I look forward to enriching 2 days full of bird chatter. I now request Preeti to make housekeeping announcement.
Hi everyone. Welcome once again to the Bird Monitoring Symposium. Before we get started with the day, I have a few housekeeping announcements for everybody. I hope that everyone who's signed up for the symposium is on Discord and that you're participating in the text-based interactions on Discord. Please browse through the different channels which appear on the left panel on Discord and post an introduction to yourself and keep an eye on the announcement channels for all the upcoming sessions and activities during the 2 days of the symposium. Also, please keep an eye on the schedule on our website. There may be some minor changes or updates that happen as we go along because of some logistical reasons. An important thing is to please go and read the code of conduct on Discord. It has been put in place thoughtfully by some of the members of the organizing committee, and it's important that we all abide by the code of conduct during the 2 days of interactions. As I said, the session links will be posted on Discord in an appropriate channel. The channel names are kind of self-explanatory, so you will be able to find your way through them. You can see that only the day 1 channels are live right now. The day 2 channels will come live after today's end of the day today. To keep the discussions organized on Discord, our volunteers will help us. All the discussions will happen in threads in each of the Discord channels. There will be a thread for every speaker that is created by the volunteers. So please post your questions to our speakers in these threads. You can use the @ symbol on Discord to tag speakers in these threads and ask your questions. We have two really interesting sessions where people will present their posters, and these are in the afternoon every day. We have allocated a separate time slot, so all of you can browse through the posters in the afternoon and post your questions. You can also asynchronously browse through the posters later after the end of the day or in between sessions during lunch or tea breaks and so on. But the participants will be there live on Discord for a text-based interaction during the allocated time. We have two very exciting evening plenaries, like Monica said. These are live talks by two speakers from outside of India. So please stick around in the evening. Today, the evening plenary is at 4 p.m. and tomorrow at 4:15. If you need help finally navigating Discord, go to the Discord help channel and please post your questions there. And finally, I hope that you have a fun and productive time at the symposium, and see you again in more interaction on Discord. Thank you. I now hand over to Vijay Ramesh, who will be the moderator for our first session for talks today. He'll tell you a little bit about the upcoming mini symposium for the day, and over to you, Vijay.
Thanks, Preeti. Very, very excited to introduce the first mini symposium for today's morning session. So the symposium will be on passive acoustic monitoring and how this tool has emerged as this transformative tool for biodiversity monitoring, especially over the last couple of years with advances in technology, and now we can essentially use recorders that fit in the palm of our hands and record data for hours and hours together and better understand biodiversity at a given location. So we have an exciting set of speakers who are going to talk to you about how they've been using passive acoustic monitoring in their research, ranging from the impacts of restoration on biodiversity to characterizing long-term impacts of human activity on biodiversity. So without any further ado, I'll hand over the session to the first speaker for today, which will be Dr. Anand Krishnan. Dr. Anand Krishnan is a professor at IISER Bhopal, and today he'll be talking to you about community bioacoustics. And specifically, he'll be telling you about the patterns and pitfalls of using behavioral traits to monitor birds.
Hi, everyone. Welcome to my talk at this bird monitoring symposium, and thank you to the organizers for having me. I'm going to be talking to you today about what I call community bioacoustics, which is using acoustic monitoring to study entire communities. I'm going to give you a very brief overview of some of the work that we've done, but mainly I want to use this talk to introduce this mini symposium and also to motivate some of the other talks and give you an idea of what the promise of bioacoustic monitoring is, as well as the potential pitfalls that you may face. Now, my lab is broadly interested in using acoustic monitoring to study community ecology. In essence, we're saying, can you use animal sounds? And these sounds are very diverse. We largely study birds, but we've also done some work on bats to obtain information on species distributions and changes. And we obtain data on these sound-producing taxa and census them and use them to examine diversity patterns. This is something that we call community bioacoustics, which is a review we put forward about 2 years ago with a prescription on how to census data, identify data, collect the necessary behavioral data beforehand in order to work on things, and actually use this as a framework to detect long-term ecological change. The review is published in Frontiers, and I thoroughly recommend you go and check it out. And we've used this in the past to look at both spatial and temporal change in communities. Temporal change is one of the earliest questions I was interested in when I started my lab. And this was something I did during the year 2017, and it was published in 2019. So you can also go look up the paper. And because I worked in a tropical region in Pune, Maharashtra, where I was based at the time, I worked in an area which was a semi-arid scrub grassland, which underwent dramatic seasonal changes between the wet monsoon season and the dry season. And in the dry season, this area also, like most tropical regions, receives an influx of winter migrants. So I was interested in how these songs change the acoustic community, the sum total of all vocal species in a landscape. And I was also interested in whether this could be used as a monitoring framework to detect the arrival of winter migrants. And what I did was I actually recorded a lot of data, censused it, and using these census data, identifying every species vocalizing in a 5-minute time window, I put them together and calculated, and what I call an abundance index, but it's not true abundance, it's actually acoustic activity or vocal activity index. And you can see there are these black species here that are represented by the black lines. These are species that were detected equally in both seasons. But there were a bunch of species noted here in red, which actually became less active or disappeared from the site entirely in the winter. And these are resident species like cuckoos, weaver birds, and painted francolins, all of which went quiet in the winter season. On the other hand, a number of species that were rare or absent in the monsoon recordings became much more common in the winter. These are represented by the blue lines. Many of these were winter migrants like lesser whitethroats, red-breasted flycatchers, humes, and greenish warblers. But others were resident species that locally moved into the area in the winter, like scaly-breasted munias, which probably follow grass seeding events. So this change in the acoustic community could be detected using simple acoustic monitoring. And acoustic monitoring can be done a number of different ways. You can put out automated recording units, which you're going to hear a lot about in subsequent talks. But you can also do this just by making long duration recordings with your microphone. And with a good design strategy, getting enough data to be able to answer ecological and behavioral questions, which is what we've done here. And in some of the talks forward, you're also going to hear about how hybrid methodology can give you valuable community ecological information. And that community level information is particularly critical for monitoring. I think in particular, you will like Abhijith's talk on mixed species flocks, which uses a fairly similar framework. We've also used acoustic monitoring to study spatial change or spatial differences in community structure across grasslands, which are a prime focus of my lab. And Shatirtho, who is now a PhD student at Minnesota, and Nafisa, my undergraduate student, in particular worked on the differences in acoustic communities between wet and dry grassland habitats. For wet grasslands, we work particularly in the Deering Wildlife Sanctuary in Arunachal Pradesh. And dry grasslands, we work primarily in Tal Chhapar, which is a semi-arid grassland habitat in Rajasthan. And using passive acoustic monitoring, Shatirtho and Nafisa were able to detect a number of species in our recordings, both in the wet and dry grasslands. And in particular, many of these species are very poorly known. So in the wet grasslands, you have a lot of rare and threatened species that we've been able to establish a monitoring framework for, like the black-breasted parrotbill, Jordan's babbler, and marsh babbler, and Graminicola, the Indian grassbird. But we've also got, in the dry grasslands, you've got species of more semi-arid habitats and sub-desert habitats like sand grouse and larks and the rufous-fronted prinia, which is a specialist of these kind of semi-arid scrub habitats. So we've managed to establish frameworks to record, detect, and census species in both habitats. And these are diverse calls that were recorded within our data set. So they're distinct from each other. They're very easy to identify. And we're in the process of accumulating a larger data set to make the analysis more streamlined. But at the outset, it required a lot of laborious censusing of data and hand annotation of data in order to make sure that the data were correct, and then to feed this into something that in future will be a little more streamlined. What we found in this case is that the two spatially separated biomes are actually convergent in terms of the structure of the acoustic community. Each species here more or less has a counterpart in the acoustic space of the other community. And this results by convergence within acoustic signals. If you want to know more about that, the paper is out and you can read more about it. But as a monitoring potential, I just use this to illustrate that you can actually monitor spatial differences as well using passive acoustic monitoring. And what we've learned over the last 6 or so years of my research in acoustic monitoring is that grassland communities and acoustic communities of birds in general are spatio-temporally dynamic. Each species is replaced by another across seasons or across regions in space. And you can actually use recorders and census data from recorders as a way to detect these differences across diverse acoustic communities. So what is the promise of this method? And that's something that I'm going to use to introduce the rest of the symposium to you. The promise of this method is it allows you to sample multiple sites at once, which allows for very efficient sampling design. You can put multiple recorders in different places and record data simultaneously. You have the data with you for years and years, so you can resurvey or reannotate or reanalyze the data or compare it to multi-year data relatively easily with storage. But there is a caveat to that, which I'll get to in the next slide. It gives you input both on single species like the black-breasted parrotbill and the Jordan's babbler, which we've been working on, but it also gives you entire community level information using concepts such as signal space approaches or community diversity approaches as well. And if you analyze your data quickly using either automated frameworks, which will take a while, I think, to get to where they need to get, but using subsampling of your data, you can pretty rapidly detect community turnover and community change in different land use types, which I will point you to Antara's talk later on in the symposium, where she's going to talk about the same thing. But most importantly, acoustic data are a behavioral trait. They are produced by birds to attract mates or to defend territories and for other purposes. And these behavioral data can give you a lot of information on activity patterns, on vocal repertoire, and other features of an animal, of a bird or any animal's behavioral suite. And these behavioral data can be used also in an ecological context to obtain information on seasonal movements, distributional changes, responses to anthropogenic activities, etc. And thus is a barometer of change that transcends disciplines in ecology as well. But bioacoustics and community bioacoustics in general is not without its pitfalls. Using this behavioral trait as a conservation tool comes with several caveats that it is very important to remember. First of all, acoustic monitoring does not provide an inventory of all species found in an area because not all species produce sound. So if your goal is to produce an inventory of biodiversity in an area, you want to combine this with other methods and not rely solely on it. Secondly, a lot of studies in bioacoustics use indices on sound recordings as a quick metric of biodiversity in an area. And I tested these indices on my 2017 data set. And you can see in the monsoon here in red, all the index values are higher than the winter values, which are in blue. But when I actually census bird diversity, the bird diversity was not different. So these indices are giving me inaccurate results because in many tropical regions, bird activity is seasonally variable. Birds sing less in the winter after the breeding season is done, and they sing a lot more during the breeding season. So as a result of these differences in activity, you can get inaccurate results from indices. So I have a rather strong and controversial view on this, which is that the indices simply do not work and should not be used for these purposes. And they are not as valuable as census data for many reasons. So community bioacoustics is really not the same as soundscape ecology. It is doing something extremely different. And I believe our analytical framework is a much more rigorous way of addressing the question. The problem, however, is that if you're not using quick and dirty analytical methods, it does result in a fairly laborious analytical task, which means a lot of data. So I would suggest not collecting as much data. A lot of people collect thousands and thousands of hours of data and use this as an advertising feature of their bioacoustic data sets. For birds, we've generally found that if you record 3 hours of the morning singing activity, you will get all the bird diversity. There are no new birds that come in later on in the day to record. So you can record 3 hours of data per day and get all the information you need, and is much more practical to deal with. And you don't have to spend insane amounts of money on hard disks to store the data, which, by the way, is not always practical for people working in a country like India. So I would say design your data collection to be relatively small, and you can analyze your data relatively rapidly using subsampling methods like the way Antara has done, which she's going to talk about in her talk. Also, what we've learned over the years of working in bioacoustic monitoring in India is that it's important to pick your species carefully, i.e. it is important to pick a species that actually regularly produces vocalizations and that you can regularly detect using vocal activity. Otherwise, you won't get enough detections to actually determine anything meaningful. And secondly, it is important to pick your habitats carefully because in India, unlike in many other countries, noise and human activity are a perpetual feature of life, and this can really complicate recording good acoustic data. So pick your habitat carefully. Acoustic monitoring cannot just be employed anywhere. All right. And finally, I will leave you with there are several analytical hurdles to consider. Automation of analysis is progressing, but it is still not where we need to go yet. And in order for automation to progress, you still need to collect the data, spend some time in the habitats, familiarize yourself with your species of interest, and record their entire vocal repertoire and familiarize yourself with it in order to progress. So there is still a lot of work that is required beforehand. There is extensive preparatory work required before an acoustic monitoring study to characterize the behavior of all organisms that you want to monitor. All right. We are still I think automation tends to work better with repetitive sounds that are stereotyped. See Swayam's talk a little further down on the lesser florican for more on that. But by and large, when you have species with a lot of individual variation, you need to actually capture that individual variation. And you also need to understand what distance to place your recorders at, which requires you to use sound pressure level calibrated speakers and experimentation with acoustic sound pressure levels to determine what the appropriate detection distances are. Okay, it is very important to remember recorders are not camera traps, and the same sound can be picked up on two recorders at once. We've said this often during our bioacoustics workshops at SCCS, and I'll reiterate here again. Recorders are not camera traps. And so the appropriate experimentation needs to be done to determine how far apart to place your recorders, although you can just be conservative and place them really far apart, like we sometimes do. And it requires you to spend a lot of time familiarizing yourself with your study species, their sounds, their behavior, and their vocal repertoire, if you want to eventually be able to streamline and automate your analysis. So there are several pitfalls of bioacoustic monitoring and caveats to keep in mind. But there are also several promises to it. Mainly, although it is not a silver bullet, it does actually enable you to sample efficiently in multiple sites at once without observers being present. It enables you to save on manpower, and it enables you to store the data if you collect your data practically and not unnecessarily, to be able to analyze it in the future. And with that, I will end my talk and lead further on to the rest of the talks of the symposium. I would like to thank Shatirtha and Nafisa, my students who did the grassland work, my funding sources and my institute, and the forest departments of Rajasthan and Arunachal Pradesh for permits and logistical help. And lastly, I would like to thank you for your time. And I will be happy to take any questions you may have. Thank you.
Thanks for that wonderful introduction to the symposium, Anand. That was a really great talk on community bioacoustics. We have a couple of questions that are coming up in the Discord channel. I'll read them out to you. First one is from Ram. He's asking, how does call count method using no devices but just listening differ from bioacoustics? Does bioacoustics mean the use of sound recording tools as a critical element?
Absolutely not. So the call count method is very much a feature of most bioacoustic monitoring tools and is extremely valuable. And I think in places where devices are expensive, although they're getting cheaper, call count methods are extremely valuable. And they're as valuable a method of acoustic senses as any of the methods I just talked about. I think call count methods are very important. I think the advantage of a call count method over a bioacoustic monitoring technique is the presence of an observer. But simultaneously, the disadvantage is also the presence of an observer. So the presence of an observer carries with it certain corrections and certain abilities to navigate difficult noise situations that you don't have with an automated recording unit. But at the same time, the presence of manpower for call count methodology makes your study a bit more expensive and puts in a bit more human effort. So those are things to be weighed and considered.
Thanks, Anand. Next question here from Piyush as well as Vishal. Piyush asks, how effective is acoustic monitoring for an urban setting? And Vishal is asking if you could give a couple of more examples of the behavioral insights you've come across from your field sites in Arunachal and Rajasthan.
Behavioral, okay. So I'll start with the first question. Acoustic monitoring in an urban setting, my 2017 study was conducted in an urban setting. It's feasible, but you have to be a little careful because traffic noise is a complete pain when you're dealing with urban environments, as is construction noise. And that can really make any sort of census really difficult. So possible, but pick your sites carefully is what I'd say. As far as behavioral insights go, we've got a lot of it actually. So we've gotten a lot of information on activity patterns and seasonal movements, and in particular for bats, which we didn't have earlier. But for birds, we found activity patterns, vocalizations, and relative uses of vocal repertoires that we didn't know before. We found associations of species that we didn't know about before, just to give a few, and just distributional data. And in some cases, we found that these species are a lot commoner than was previously supposed, because you can't see them, but you can definitely hear them, if that helps. And if there's any more, I'm happy to answer questions in the future. Although I'm having trouble with Discord, you can always ask me questions and I'll get an answer to you.
Thanks so much, Anand. There are a couple of other questions on Discord, but for the sake of time, we'll have to move on to the next speaker.
That's asking.
We now have Hari Krishnan, who will be talking to us on monitoring an endemic bird or endemic birds from the Western Ghats. He's specifically going to talk about using bioacoustics to investigate the seasonal pattern of bird presence. Hari is a student at IISER Tirupati. Over to you, Hari.
Hello everyone, I'm Hari. I'm currently working with Dr. Robin at IISER Tirupati, and I'll be talking about how bioacoustic can be used to study seasonal patterns of birds' presence. So, seasonality studies in birds. So, why is it so important to study seasonality? Because seasonality can give various insights about birds' responses to environmental changes, given that there's a lot of anthropogenic changes that's happening, and understanding how species respond to these changes is of vital importance. Also, it can give patterns about breeding and migratory things. Also, seasonality studies can also help us understand how species are using habitats, different habitats. So basically, habitat usage can give various insights about habitat usage patterns. And so studying such seasonality works requires long-term data, which is basically you have to go to field and collect data for longer periods of time. But this comes with a expense. So this can be a very labor-intensive task because we have to go to the field for longer periods of time and get data for longer periods. So this can be an expensive task as well. So, so another alternative for such labor-intensive task is to make use of bioacoustics. So bioacoustics is basically using autonomous recorder units to collect data about ecosystems. So yeah. So autonomous recorder units, which is basically we can deploy for longer periods of time, and we can use this to record data for continuous period. So the data can be very continuous without much human intervention. So it's a non-invasive way of sampling where we're not in the field disturbing the animals, and also there's a permanent record of data along with very fine scale continuous data, and it's less prone to observe bias because the recorders are collecting the data. And also, it's a very easy, good method to study a highly cryptic and rare species which are often undetected while we are doing field sampling, when a human observer is doing a field sampling. And so far, bioacoustic has been used for biodiversity monitoring, behavioral changes, to study behavioral changes, animal movement patterns, and habitat use, and species responses, etc. And only thing is that species have to be vocal to use bioacoustics, and also, since we are collecting data for longer periods of time, there's a huge amount of data that's been collected mostly in GBs and TBs. So storage and processing becomes a challenge, but use of modern technologies can ease this. So the current objective of the study is to see if bioacoustics can be used to detect the seasonality in presence of birds over a long period. And such studies have already been done in the temperate landscapes, temperate habitats, but not in a Western Ghats tropical landscape. So it's important to see whether this works here also, because our Western Ghats landscape very acoustically rich and very rich in biodiversity. So there's a lot of vocalization that's been happening, and often the recordings that been made using these recorders, autonomous recordings, are very complex. Like a lot of species are vocalizing at the same time. So it's a very challenging recording. So to detect the seasonality, we need to be sure of the species that is seasonal. So we decided to choose large-billed leaf warbler, which is a winter migrant, which migrates to the southern India during winter, and it breeds in the Afghanistan to China and Himalayas during the breeding period. So we know that the species is seasonal in the Western Ghats in the southern India. So this is the weekly bar chart of eBird occurrences from the species from our study site, which is in the Vindigal district of Kodaikan, Vindigal district of Tamil Nadu. So you can see that the species is seasonal. It is not present in June, July, August, and September. And September it starts again. You can see the seasonality in presence and absence. So yeah, this is another weekly chart of Tamil State, Tamil Nadu, where you can again see the seasonality, and also this is the chart from Kerala where you can see the seasonality, but there is some presence which is observed in June, July, but still from on an average we can see that this species is present. So we expect the same thing with our aquatic data, So in order to study something like this, we deployed recorders in the Palni Hills landscape. So our study site was the Palni Hills of West Bengal, which is a Shola skyline landscape. So this is actually a part of another study where we look at how birds are responding to exotic timber plantations, which is again a threat to the Shola skyline landscape. So we deployed recorders in different plantations, which is old plantations, and intermediate old plantations. Also in Shola forest, which is again a control for our detection for the study. So yeah, so we deployed these recorders from July 2018 to June 2019 across eight locations, and all the recorders were deployed like about 1 to 2 meters above ground to record most of the acoustic environment, and it was also placed 100 meters away from road and 500 meters away from Shola forest in when it comes to plantation forest. So yes, such things were taken into consideration, and this is the picture of how recorders are deployed in the field. And yeah, the few more pictures where you can see that the recorders we deployed on a tree trunk. So yeah, so what happens after is that recording schedule was basically recording for 5 minutes in every 10 minutes, so across 24 hours across 8 locations. And so every cycle was around 30 days. So after 30 days, you have to go to these 8 locations and collect data and change batteries. And so each recorder collected about 100 GB of data per cycle, and 60. So recording at 16-bit 44,000 sampling rate. And we format, since we're interested in birds, so this was okay for recording birds. And we collected around total 8 TB of data, which is basically 25,000 hours of acoustic data. So after collecting so much acoustic data, which is in TBs, we cannot manually annotate this data just like that. So we have to think about various automated detection algorithms for detecting these species from these recordings. So as I told, this is a part of another study. So we had on a lot more species to detect from the recordings. So when it's impossible to do manual annotation, we tried to do various automated detection algorithms. For example, in Raven Pro, there are some in Kaleidoscope software. There's another automated detection algorithm. So there are R packages to do. There are AI based things like CNN based neural networks to look at to do automated detectors. But what happened is that for all the other species and for this species, the automated detectors didn't work. It didn't give a good precision and recall. So we decided to check the automated detection part too, but then do on manual annotation. But then we cannot use the entire ATB of data to do manual. So we decided to subsample the data based on peak vocal activity of the bird. So we found that the peak vocal activity of birds are from 6 a.m. to 9 a.m. And we decided to subsample those data between 6 a.m. and 9 a.m. So we checked the other daytime data, and also we decided to only take from every alternate day, because it's again a huge amount of data to manually annotate. So we had around 1,500 hours of acoustic data that is sampled across years from all the eight recorders. So we used another assisted method for manual annotation, which is again using a software, Kaleidoscope Pro, which is, so they have a particular feature called cluster analysis, where the similar looking spectrums are grouped into clusters. So we have, for example, if we have large leaf warbler, all the large leaf warbler similar calls will be clustered in one cluster. So it's easy to manually annotate rather than in Raven, which we go where we go and throw boxes and around. So I'm going to play this call. So our target call for large leaf warbler was this PP call. So we manually annotated these calls for large leaf warbler, and then across 8 locations for all throughout there. Then we got the presence absent matrix of these. So coming to the results part of this talk. Yeah, so we had 8 locations and 4 different habitats, that is young, intermediate, old, and shola. And we clubbed the 2 youngs together, and intermediate and old and same shola, like we clubbed all the 2 locations. So this is the clubbed results of from all these habitats. We can see some seasonality patterns, where here you can see that the green indicates the presence of species and the red indicates absence of the species. So we can see that in the months like July and August, there are some absence of the species as expected from the eBird records. In eBird, we can see that in July, August, and May, June, in most of the places, the species are absent. Only in Kerala there was some presence, because sometimes these winter migrants choose to stay back, and so in our case, in these when we were doing manual notation, we could see that in July, August, May, and June, the detections were very less, almost 1 or 2 or less than 5 max. So we can see that another important thing to notice is that we can use bioacoustics to also detect such fine scale data. We were able to detect even one call in that month. So yeah, so this is the seasonal pattern of large-bill leaf warbler. And here now we can talk about the vocal activity patterns of these birds in these 4 different habitats. So vocal activity, number of calls detected per species in every month. So we can see that as the wintering time starts, that is basically in September, the vocalization increases, and it peaks around October, November in all the habitats. So this is because mostly when species arrives and try to different territories, and they try to do they tend to more vocally active at that time to different territories and create social bonds and everything, etc., with other species and within species. So that's why the vocalization. This is actually not our finding, but few studies have said that winter migrant does that, and the vocal activity gradually decreases across all, and decreases, and it's kind of absent in May, June, and July and August. But there is some present, which is basically maybe some species choose to stay back or something like that. So we can say that bioacoustic technique can be used to study seasonal patterns of bird presence, also to understand vocal activity patterns of birds for longer periods of time. And so this is not the end, actually. So we can do a lot of other stuff with this data, with this presence, absence, and vocal activity data. We can model these presence, absence, or vocal activity data with other variables to give some other insights. For example, we can model this with temperature data or humidity data, to see how vocal activity or presence, absence varies with varying temperatures and humidity and various other factors. So overall, I think we can use bioacoustics techniques to study species in the Western Ghats, especially in the Western Ghats, because it did patterns, and we were kind of sure that this is patterns. Yeah. So yeah, and I would like to thank Tamil Nadu Forest Department, Kodaikanal International School, ISO Tirupati, Jobin, Aravind, Zanish, Chiranjeevi, and field assistants, all the members of Eco Lab at ISO Tirupati. Yeah. I think, yeah, thank you. Thank you.
Thanks so much, Hari, for that wonderful talk and such insightful information on studying a single species and learning so much about its seasonal presence.
Thank you.
There are a couple of
for that wonderful
talks on Discord. I'll read them out.
Information on studying a single species and learning so much about its seasonal presence.
So the first question is from Preeti. Preeti is asking, how did you arrive at your recording schedule? What were the considerations in general when coming up with a recording schedule? I think we've lost Hari briefly.
Oh yeah, sorry. It's. I think the YouTube was on somewhere. So yeah, I think yeah. So we did try various combinations of recordings. So I saw the Preeti's question, and then like we did record for like different amounts of time, and and checked how all those are working. Also, we also looked at how also the logistics part of these recorders. So basically when we decide a schedule, we had to look. So we looked at how how long the recorders are running in the field. And so we since our field work was in Kodaikanal and we were in Tirupati, so we had to go back like all the time. So we needed like a big chunk of sorry, not big chunk. So it's a long. So we wanted to record for long term. So so our recording schedule, we decided based on how often we can go to the field. So this was like every month we were going to field to change the battery. So and also based on the other eight. So actually this is a part of another study where we are looking at presence absence of other species as well. So like 5 minutes was a good amount to detect and not detect these all the other species that been studied also. And this wasn't back in 2018. So back then there was not much research available to the present time.
Right.
But yeah, if you ask me right now, I don't think I'll go for 5 minutes or more than. So I may go like every 1 minute in a 10 minute or something like that, much lesser. I think this was a bit of oversampling than right now. So I don't think we have to use 5 minutes right now, but so I think I can type more on the Discord.
Sure. Yeah, thanks, Hari. A couple of other questions on more on the detecting the large-billed leaf warbler itself. So is there a chance that they're actually staying back and you're missing their vocalizations during the monsoon season, possibly because they're not vocalizing, or are they being quiet during these months?
I mean, that's definitely a chance, because we don't know, since we don't have a ground data, so we didn't do any ground truthing for this. So it was just the recorders that been running. And so all we are dependent on is from the vocalization, based on the vocalization. And we know that from other records that winter migrants do stay back, not all, but some. So I don't think we can comment on that from this current data set. Yeah, but yeah, definitely they can stay back. And if they're not vocalizing, we'll definitely miss it.
Sounds good. A couple of other questions on, have you already carried out any analysis that looks at correlations with temperature or seasonality or weather?
No, I haven't done that, but I'll be doing it. So I'll be giving another talk in ATBC most probably. So in which I'll be talking about how seasonal patterns are in much more detail. This is just an exploratory thing. I just looked at large-billed leaf warbler just to show that it's seasonal, and bioacoustics can detect the seasonal patterns on a larger scale. So I'll be in the ATBC talk, I'll be talking about how other species are using these habitats for longer periods of time. What are the variations there, how the seasonal pattern varies with temperature, and also, but not in this current study. Yeah,
sounds good. Thanks so much, Hari. Again, we'll have to move on to the next speaker. The next speaker is Abhijith, who recently finished his master's from NCBS. Today he'll be talking to us about acoustic traits and community ecology of mixed species bird flocks.
Good morning, everyone. First of all, I'd like to thank the organizing committee of the Bird Monitoring Symposium for inviting me over this talk. And the small talk, I'd want to emphasize how we were able to add another dimension to our understanding of the community ecology of mixed species bird flocks by incorporating acoustic data. Mixed species bird flocks, as they are known, are generally defined as foraging groups of birds that are composed of 2 or more bird species, and they are known to greatly benefit from protection from predators and foraging efficiency from being part of this group. But at a global scale, what we know is that there is a general propensity of these flock participants to group together with other similar sized species, and this is attributed to the enhanced dilution effect that is accrued by them, because similar sized species grouping together would mean that they share similar cohort of predators, and thereby the risk of predation is equally spread across all participants, because of which there is also relevant social information in the system. But what do we know about communication in mixed species bird flocks. Most often, flocks are detected not visually, but because you hear a lot of birds calling together in short proximity to each other. And they're quite vocal, and I'm just going to play a small recording here. As you can hear, this is a flock recording, and a lot of birds are simultaneously calling together. And vocalizations are also an integral part of the life history of birds. And in the context of mixed species flocks, we also know that the calls provide anti-predatory strategy, there is eavesdropping calls, there is intraspecific signaling, and there is predation risk assessment that comes through these vocalizations. But building together all that we know of flock, we know that similar sized species group together, but body mass and related characteristics are also known to be correlated with acoustic parameters of species in such a way that the larger bodied species can generally produce lower frequency ranges as well. But if this correlation translates to acoustic similarity among the system, what we end up, or what the birds end up having, is a cost that can come about just because they are masking each other's information. So the more overlapping acoustic features the birds share, the more likely they are to mask each other's information. Now thinking about this, we wondering how it is that the birds might be able to overcome this particular dilemma. And being heterospecific groups, the benefit of species is that the participants do have the flexibility to maintain similarity and yet maintain dissimilarity across different trait axes. Just to give you an example of how this can be done, the people have shown that the birds tend to group together with similar size species, but it could also contribute in terms of cost because there is also likelihood of overlapping resource uses. And thereby what they find is that by being similar in body mass, the species are being more distinct in their foraging patterns and behavior. So similarly, this could also be attributed or connected to how birds might be able to decouple morphology and acoustics in these flocks. And our basic aim was to understand the relationship between acoustic and morphological trait space across different flock richness classes. The study was conducted in Wayanad Wildlife Sanctuary, which is nestled within the larger protected area network of Nilgiri Biosphere Reserve. So this is how the general habitat looks like. It's relatively open, allowing us to have a good visual access to the flocks that we are following and take notes on the birds that are probably calling. In terms of data collection, we followed transit sampling, and each encountered flock was acoustically recorded and visually noted down in terms of the number of or participant species that were part of the flock. Now, talking about recording as a process here, I consider it somewhere between the active and passive. So when we have a clear idea about what we are recording and we are following it, it becomes active. But here, the scale of following the system is at the level of a flock. So we know that we are following a flock. At the same time, the recording is also open to other species that are neighboring to these flocks. So to ensure that this does not interfere with the analysis of the data and data collection, I used to speak into the recording whenever there was an occurrence of species vocalization from outside the flock system itself. Now that we have a visual census data about the participation, that was translated to a flock metrics, and each species here was considered a participant only if the participation was maintained throughout the duration of the data collection. Now, this was very integral to data collection because unless we have a reference list of participant species, we would be unclear as to what to look out for when we do census on the acoustic recording that was obtained from the flocks. Now, what we have here is we were able to track changes in species composition across different flock sizes, and the morphological traits and the data about it was available in an open data source, which is A1net. Coming to acoustic recordings, all of it was done using a Sennheiser ME66 directional microphone and a Zoom H6 recorder. To sample these recordings, that is to undertake census along the recording, we followed a dispersed subsampling method, and this was to ensure that the entirety of the recording was equally sampled or evenly sampled. So consider this block that I've shown you as a 7-minute recording, and we would be sampling every other block, that is the alternate minutes would be sampled. And the census would be conducted on these time frames using the visualizations provided by Raven Pro. And we could convert this data into another acoustic census data, in which the sampling unit here is the time frame. Now that we have all the information, we were able to extract vocal activity index of each species. Looking or thinking back at the matrix that I just spoke about, we would have a clear number of time frames that in which the species had vocalized. And we also know the time frames in which the species was a participant in the flock. And a basic division of it gave an index, a probability of each species being vocally active when it is indeed part of a flock. Now, the rest of the parameters was extracted using Raven Pro software. And to just give you a broad outline of how the screen looks like, these are the graphs that are seen, and this is how it's visually represented. You have calls, and here this is a graph which is called spectrogram, and on the x-axis is the time lapse, whereas the y-axis provides the frequency. And we were able to carefully extract 8 acoustic parameters that were robust to variation in amplitude and subjectivity of note digitization. And to broadly give you a sense of what it does is that by extracting these critical parameters, we were able to define the shape of the calls produced by the species. Now, to use a particular value for a particular species, to associate that, we ran PCA on these variables, and the first PC1 score explained around 80% of the variation, and the average PC1 was used for the further analysis ahead. To broadly sum up the analysis, we have observed data, which is the species composition matrix we spoke about. And we have also extracted the phenotypic traits, such as the morphological data and the acoustic data. And all the one that is there in the presence-absence matrix could now be replaced by these particular morphological and acoustic traits. And the entire matrix can be reshuffled and randomized 1,000 times. And this was done by maintaining the flock sizes as the same and pooling species based on its probability of occurrence in the entire matrix, that is the community here. Now that we have the null data set, the variation of the traits were captured by calculating the standard deviation, and this could be compared to the null data set, because we could now have a 95% confidence interval, and which detected what can be expected by chance alone. Coming to the results, in general, the flock system that we captured was dominated by smaller sized birds. As you can see, towards the left side, you have more representation. And when plotted body mass against the phylogenetic relatedness of bird species, what you can see is that to the left here is that the smaller bird species are relatively closely related to their larger counterparts. And when we look at it from a vocal activity index from the right, what you can see is that the same group of small bird species which are closely related are also relatively more vocal. Now, what does that tell us? You have a flock system dominated by smaller sized birds, which are closely related and which are also very vocal, which makes it important for us to look at whether there is a possibility of them masking each other's information, because there is a lot of public information generated. Now, coming to another set of results, on the x-axis here is from the left to right, small, medium, and large flock richness classes, so as the flocks grow. And the first two categories, that is small and medium groups, they constitute around 87% of all flocks. And the standard deviation on the y-axis here is the body mass variation. And as you can see, the whiskers are the 95% confidence interval, and the orange dots are the variation that was observed in the flock. And they seem to be more similar than what can be expected by chance, gently suggesting that it is indeed that the birds are grouping together with other smaller sized birds. Now, what do we know till now? There is body mass similarity among birds, and they are generally dominated by smaller sized species, which are closely related and relatively vocal, making it important for us to look at the acoustic similarity patterns. Now, when we ran the same analysis using the PC scores that were available, what we starkly observed is that the participant species are not similar in their acoustic signal parameter space. As you can see, all of the orange dots fall within the 95% confidence interval, suggesting that there is some form of deep coupling between the traits, irrespective of their correlation. Just putting it side by side, to the left is the body mass similarity patterns, and to the right is the acoustic PC1, where one is more clumped than expected by chance, whereas in acoustics, it is what is within expected by chance. And this provides us evidence in some way to suggest that there is some form of decoupling among these traits, irrespective of their correlation, and possibly allowing species to maybe overcome the cost of masking each other's information as well. So in summary, the initial results suggest that species go together with other smaller other similar sized birds, and it generally more pronounced in the smaller sized birds, which are closely related and vocal, and they are acoustically non-similar. Now, the study wouldn't have been possible without the support of a lot of members, including the institutional support by NCBSTIF or NCF, my committee members who guided me, Preeti, Samira, and Anand, my dad, Madan, who's my field collaborator, Arul and Badusha for helping me out in the field, the entire NCBS and Wildlife batch, family and friends, Kerala Forest Department, and thank you all for listening.
Thanks, Abhijith, for that wonderful talk. It's always fun to learn more about mixed species flocks, and especially about their vocalizations and communication patterns. Karthik, Prachi is asking you a question. How did you select the area for deploying passive acoustic monitors? Do mixed species flocks forage in the same area for you to deploy the units in advance?
Yeah, so we did not deploy it before we were actually collecting the data. So we used to follow the flocks actively, and that's why I mentioned that it falls somewhere between active and passive recording, because in some sense you're following the flock, but there is a limit to how much you can actually follow the species and which ones are vocalizing. So that is where the passivity comes into it. So it's somewhere between active and passive recording. Yeah. So we did not deploy anything. We were actually actively following the flocks.
Got it. Thanks, Abhijit. Gaurav's asking that you did choose around 8 parameters, but there could be other parameters which may be similar and are not chosen in the study, right? How did you control for this?
Yeah, I think similar to what Anand had said, there was a publication that came out, and the point was that you select the parameters which are probably not susceptible to changes because of the change in amplitude and probably the direction of the microphone, but the ones that are robust to these things and which in totality would define the shape of the call. So if you look into the parameters that we've chosen, it clearly outlines the shape of the call in totality, and that was the reasoning behind it.
Got it. While we wait for other questions, I have a question for you, Abhijit. So I know you mentioned something about smaller sized birds being more vocal. I wonder if that pattern ever changed if there was a predator around. Was it often the smaller sized birds vocalizing a lot more, or was it larger sized birds?
The study that we conducted was particularly focused in all non-alarming contexts, because from the literature we know that the vocalizations are more or less converged in that context, so that there can be the information is more or less available for other species as well. But it could well be that during that context, most of the species in the flocks do vocalize together because the context is very different from that of foraging. But in general, overall, what we see is that the birds that are closely related, which are also passerines and songbirds, which in general tend to be more vocally detected as they flock.
I see. Okay. Thanks again, Abhijit, for this wonderful talk. We are now going to move on to the next speaker. That's Amrutha Rajan. Amrutha is going to talk to us about establishing a detection framework for the endangered forest owlet using automated recording units.
Good morning, everyone. So I'd like to present my work on detection framework, developing a detection framework for the endangered forest owlet using automated recording units. As we all know that documenting biodiversity is essential for the future conservation and long-term monitoring of the species, especially for threatened and endangered species. So we know that traditional survey methods have been replaced rapidly with the novel technologies like bioacoustics. And how efficient are these bioacoustics tools in detecting let's say a rare vocalizing species such as Forest Owlet, and maybe difficult to detect a species which is difficult to detect with infrequent human visits. And so the development of ARUs and associated algorithms, the signal detectors, have enabled researchers to conduct bioacoustic deployments, long-term bioacoustic deployments, which generate large data sets and that cover large landscapes too. So here we took a Forest Owlet as our study. And we know a Forest Owlet is an endangered bird to central India that was thought extinct. It was recently discovered after 113 years. So in this particular study, we used to ask our main focus is to test which detector works better in detecting Forest Owlet calls, and how far can an ARU record the clear response of Forest Owlet, and which later can use for the signal detector. And are there any variations in Forest Owlet detections when we combine with other co-occurring owlet vocalizations? So the study area we used down district area or the study site, which is the northernmost end of the Western Ghat. And we had a total of 46 grids, which is 1 by 1 square kilometer grid. So what we did to detect how far the ARU can record the bird call. So we placed ARUs, so in the center, from the center of each grid, 1 by 1 square kilometer grid, we placed ARU recorders at a distance of from 100 to 1000, let's say. And so we did the playback at the center of each grid, and we marked the response site. So from the response site, we placed the recorders accordingly, as you can see in this figure: 100, 200, 300, 400, 500, and then 1000 meters. So this is how we did the detection distance to test, how far an ARU recorder can better record the bird call and use it for the detector algorithm. So here comes the method used for how the Forest Owlet detections we planned by selecting 2 detector algorithms, as you can see, Raven and Kaleidoscope. So we used a test data set, which is 1 hour data, and a long recording data, which is a 6-hour data. So from this detection distance experiment, we collected this test data and long recording data. So in Raven, we trained the data using this test data set by creating templates, which I discussed later. And Kaleidoscope, we trained the data set using so the data set include both FO and no other bird calls, actually non-forest owlet call birds. So what we used to do in Raven, we first test this data set by selecting appropriate templates by running template detector validator. So we select the best templates and then run the long recording data to test the accuracy or precision. And for Kaleidoscope, there is a 2 simple clustering algorithm is happening. So that is based on the k-means clustering classification. So it basically so we have to train the data set in such a way that what are the forest owlet calls and what are the other bird calls. So finally, we'll run the long recordings. So we have to manually check what are the true positives and false positives. So both the field recordings, the long field recordings was used the same for testing these recorders. So these are the templates which we used in Raven for the template detector. So we basically selected these templates based on the template detector. So this is how we choose the template detector based on the F-score values. So this F-score values is nothing but it's a mean, harmonic mean between precision and recall, the F-beta score. So this F-beta score is based on the better precision recall, true positive rate, false positive rates calculated by software called detector validator. So here we can see the threshold, various threshold values and frequency ranges from 0 to 100. So here we got 0.6 not for as a better F-beta value to run the template detector, and we choose this 0.65, 0.6 to 0.65 threshold value to select the templates. So these two tables you can see is without loose, match, and stretch. These are the different parameters in Raven template detector. So basically, we tried various parameters and selected the best F-beta score template. And then here you can see the output of template detector. This is how the detection looks like, where you can see true positives and false positives. Yeah. And so how kaleidoscope? So kaleidoscope, we have to give two sets of training. So after the 2 sets of training, it will we can give the long recording data, and it will give an output like this, which will tell which are the FO calls and which are not other FO recordings. So this is the result window of kaleidoscope. And then, okay, here are the results. So coming to the results, you can see in the graph, when we detected in Raven, the Raven works better than kaleidoscope with the 10 to 27% better performance showed by Raven, and for both call and song. So this is the in right top you can see the Forest Owlet call, which is a disyllabic call, and below a monosyllabic call. So these are the 2 types of vocalizations we detected in the detector. And so when we combine the detector performance of Forest Owlet with other 2 co-occurring owlets like Jungle Owlet and Spotted Owlet, as you can see in the figure, the Raven performance got decreased by 10%, but the kaleidoscope increased in the detection percentage by 11% increase. So totally it's a contrast. So this, what is this saying is kaleidoscope with, the accuracy is getting better when we mix, with many other species calls. But in Raven, they need pure, it is getting, it will get better when we are reading better templates, suitable templates with high quality templates. So these are the templates we used in Raven. So here you can see in the A diagram, we selected the templates without a band filter, and B with the band filter. So here we'll remove the background noise, only the, only the syllable, and likewise the same for the other type of call. So when we use this method, with band filter, we got a better results for Raven detection. A 43% increase in the Forest Owlet call and song detection. So, yeah. And so coming to the comparison, the signal detector comparison, as we have seen from the diagram, graphs, that the Raven was performing better as it needs less training data. It just needs 5 or maximum 4 to 5 better quality templates from the handheld recordings. And it runs very fast. Just 1 minute, for a 6-hour data, but it takes when we comparing, when we want to analyze the true positives, false positives, that you have to manually check for both the detectors. And for kaleidoscope, it need a large number of training data, for example, 100 to 150 syllables for each call, and the detector performance will be better with the presence of, large number of syllables. And it takes around 3 minutes to run the 6 hours data. So we compare the 6-hour data for both the detector, and the Raven showed high F-score values for both the vocalizations. And in Raven, we got less false positives for many few species like spotted owlet, jungle owlet, greater vocal, whereas kaleidoscope have high false positive rate. So from these results, we can conclude that Raven was performing better than kaleidoscope, but these two different algorithms works in two different ways. So by this overall 75% of forest owlet detections from Raven, it indicates the scope of future use of automated recording units and the algorithm in detecting the species in future surveys. And by detection distance experiment, we have seen that it is better to place the recorders up to 300 meters from the response site. So once the response site is marked, then it is better to place the recorder around 300 meters to get the clear response of forest owlet. To get a higher precision when we are running the detector validator. So yeah, thank you.
Thanks, Amrutha, for that wonderful talk on detecting an endangered species and learning more about it using passive acoustic monitoring. While we wait for some more questions to pour in on Discord, I know one of your key results was that Raven tended to perform better than kaleidoscope for this particular species, the forest owlet. Would you say that in general, you would recommend using Raven over kaleidoscope, or do you think it's very species specific?
So I think it's very species specific, Vijay. So if we want to choose for a particular call or particular, vocalization of a species, definitely we can go for Raven over kaleidoscope, because kaleidoscope, it's like detection of multiple species. If you want to run a long-term monitoring of multiple species, then kaleidoscope would be best. So it totally depends on the objective or which species and what kind of goal you have. Yeah.
Got it. Thanks, Amrita. Vrishal is asking, did you detect any temporal differences in vocalizations of the forest and jungle owlets? If yes, what were they?
So meaning temporal detections, so the difference in detection over the years, or I didn't get the question.
I presume, I think Vrishal is wondering if forest and jungle owlets were vocalizing at different times.
No. So basically what we detected is there any detection variations while doing the detector algorithm? So if, let's say, the jungle owlet is present in the acoustic recording, the long-term recording, would it affect the detection of forest owlet? So it may increase the false positive. Let's say it may say it's a forest owlet, but actually it's the jungle owlet code, like that. So in the presence of jungle owlet vocalizations, more false positives you're getting in like Raven. That's what I meant when I said we also compared other owlet species presence. Yeah, I hope that.
I see. Okay. Ram K is asking, what was the maximum distance that the owlets were recorded from?
So basically, we placed recorders from 100 meters to 1,000 meters. Once we recorded the response site, so we placed the recorders from the response site. So we got only up to 300 meters. We got the detections. Yeah. So up to 100 to 1,000 meters, we placed the recorders. Yeah.
I see. Okay. Prachi is wondering if you placed these units in specific locations to detect the forest owlets. Were there any consideration in terms of where the recorders were placed?
Yeah.
So actually, we did, surveyed the 46 locations, and from these 46 locations, 16 only we got the Forest Owlet positive sites. So we only did the experiment in these positive locations. That means we are sure the Forest Owlet is there in these 16 locations. And we did all the experiments in those grids. Yeah.
Thanks, Amrutha, and thanks for your great presentation. We will now break the morning session. I think I wanted to thank all the speakers who contributed and told us more about their research in this session. We'll now take a break until 11:45 a.m. So please do join us for the second session, and definitely head to the appropriate channel on Discord, as the right YouTube link will be posted there. Thanks again, and see you all soon.